Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology Department. Welcome to online learning in Microbial Technology. Today in this session, we learn about the microbial production of microbial enzyme or the enzymes produced from the microorganisms. Enzymes are naturally occurring biocatalysts. They act as the catalyst inside the cell, which accelerate the metabolic reactions. Various metabolic activities and production of primary and secondary metabolites are not possible without the involvement of these biocatalysts, that is, enzyme. Enzymes produced during fermentation are mostly extracellular. So these microbial enzymes, their secretion may be extracellular or it may be intracellular. But during fermentation, mostly extracellular in the secretion, but few enzymes are intracellular. The example of intracellular enzymes are asperginase, invertase, etc. And like that, the extracellular enzymes, that is, they are produced outside the cell to the medium, that include amylases, cellulases, invertase, lactase, esterase, lipases, proteases, etc. So microbial enzymes are biocatalysts. They are produced from the microorganism. Within the microorganism, if it is produced, it is intracellular enzyme. If it is produced to the medium, then it is called as extracellular enzyme. And we saw the few example for extracellular and intracellular enzymes. Next, we'll concentrate on the production of the microbial enzyme. The production of the enzymes from the microorganisms takes place in following steps. That involves the isolation of microorganisms, strain development, and another one is preparation of the inoculum. The microbial enzyme production involves first and foremost selection and isolation of the microorganism, strain development, further preparation of the inoculum. Isolation of microorganism is very important, and the microorganisms are isolated on the culture media following the microbiological technique. The main aim of the isolating the suitable microorganism is to production of enzyme in high amount, the large scale production of the enzyme, and other metabolites in low amount. Only the enzyme production should be more than the other metabolite. So large scale production of the enzyme should be done by the microorganism but not other metabolites. Then completion of the fermentation process in short time. That means we should select the microorganism which will produce the enzyme in short duration of fermentation process. And another important thing is we should select and isolate the microorganism which will utilize low cost culture nutrients. So it should not require the high or very costly the culture nutrient. So then it will be very difficult for the large scale production of the required enzyme. Hence, we have to select the microorganism which will produce the high amount of enzyme, and it will produce within short duration of the fermentation utilizing very cheap and low cost nutrient. Once the suitable microorganism is obtained, its enzyme producing ability is optimized by improving the strains and formulating the culture medium. So here they'll be changing the pH and other physical factors like temperature. So that will be improving the characteristic of the microorganism. The strains of the microorganisms are developed by using mutagens. So the strain improvement technique, many techniques are there. Among those techniques, they are using the mutagen. So mutagens will be used for improving the strain or the characteristic of the microorganism for the large scale production of the enzyme. That is, they'll be using the chemicals and ultraviolet light to produce more of the enzyme. Here, after treating with the mutagens, inoculum of the microorganism will be prepared by multiplying its spores and mycelia on the broth or the liquid medium. So now you have the microorganism that is isolated and its strain is improved, and even the inoculum is prepared in the liquid medium. Now what we have to do is we have to concentrate on the media composition. You have to concentrate on the media composition. Here, the culture medium is formulated in such a way that it should provide all the nutrients supporting for enzyme production in high amount, but not for other microbial metabolites. And also, the medium formulation should not trigger the microbial growth. Vegetative growth should not be triggered. Instead, it should produce more of enzyme. For this purpose, an ideal medium must have the cheap source of carbon, nitrogen, oxygen, amino acids, growth promoters, trace elements, and little amount of salts. And here we should know whether the microorganism is aerobic or anaerobic. So that also play an important role in formulating the medium. Care must be taken to maintain the ionic strength of the media, that is the pH, during fermentation. For a specific microbe, pH, temperature, and formulation of the culture medium is standardized prior to the inoculation, prior to the transfer of the microorganism into the medium. The production of the enzyme increases with the concentration of the culture medium. And here, the carbon source, that is the carbohydrate, for the production of the more of the enzyme, usually they use molasses, barley, corn, wheat, and starch hydrolysate will serve as the carbon source for the maximum production of the microbial enzyme. Along with that, the protein source like meals of soybean, cottonseed, peanut, and whey, corn steep liquor, and yeast hydrolysate will act as the protein supplement, where they provide the nitrogen component for the synthesis of the amino acids and other enzymes and protein in the microorganism. CFTRI Mysore has developed technology for conversion of tapioca starch to glucose by using the fungal enzyme. By using the fungal enzyme. The enzymes have also been isolated from bacterial cultures, which convert the glucose to fructose in starch hydrolysate. Uh, Here in CFTRI, about 15 tons of high fructose syrup could be obtained from tapioca starch from 1 hectare land. But otherwise, if you take it from sugar cane, it is only 5 tons. It's only 5 tons. So it is almost 3 times more of high fructose syrup will be prepared in the CFTRI compared to isolating the high fructose syrup from the sugar cane. Medium should be sterilized batch-wise in large size fermenter. For this purpose, continuous sterilization method is now being used. After medium is sterilized, inoculation with the sufficient amount of microorganism, that is inoculum, is done to initiate the fermentation process. The traditional method of um, enzyme production usually that is the surface culture technique was used, where inoculum remains on the upper surface of the broth. Nowadays, they are using the submerged culture for the large scale production of um, the microbial enzyme, where the chances of infection will be less in the submerged culture method. So this surface culture technique is being used in production of the amylase enzyme from the aspergillus species, and protease from the again aspergillus, and pectinase from the penicillium and aspergillus species. Continuous culture technique is used for cellulase production by trichoderma microorganism. So in all this, they are concentrating more on the large scale production of the microbial enzyme, respective microbial enzyme. During the production of this microbial enzyme, growth condition has to be maintained in the optimum, or you have to concentrate on the pH change, temperature, oxygen has to be maintained inside the fermenter at optimum level. So these physical factors differ from microbe to microbe, and even in the same species of microbe, they will be varying. The little amount of oil is added to the fermenter to control foaming. Froth formation takes place inside the fermenter during fermentation. To avoid that, oil will be added. So this oil will act as anti-foam agent. After about 30 to 150 hour of incubation, after inoculum is transferred, extracellular enzymes are produced by the inoculated microorganism in the culture medium. As it is extracellular enzyme, it will be produced in the fermenter in the medium. Most of the enzymes are produced when the cells are in the growth phase, that is in the exponential phase of the microorganism. Usually most of the enzymes are produced, but in few cases they are produced during the exponential phase. Besides extracellular enzyme, other metabolites are produced in the fermented media. These metabolites are removed after enzyme purification. So the microbial enzyme production usually takes place at the fag end of the exponential phase, but few are produced during the log phase or during the growth phase. But during the production of the microbial enzyme, along with the microbial enzyme, many metabolites are produced at very low concentration. These metabolites will be removed during enzyme purification, because this metabolite may hinder the activity of the enzyme. When we are using the microbial enzyme or any enzyme, it should be at the purest form to perform the reaction. When fermentation is over, the broth will be kept at 5 degrees centigrade to avoid contamination, and recovery of enzyme from the fermented broth of bacteria is more difficult than from isolating the enzyme from the filamentous fungi inoculated the fermenter, because the fungal broth is directly filtered and it will be centrifuged at particular pH, but the isolation will be very difficult in case of the bacterial culture. Therefore, what they do is to make the process easy for the production of the enzyme, the bacterial broth is treated with the calcium salts to precipitate the calcium phosphate, which help in the separation of the bacterial cells and the colloids. So just by adding the calcium salts, all the bacterial cells will be separated. Then the liquid is filtered and centrifuged to remove the cell debris. So that will not be observed in case of the fungal culture. It is very easy for the separation of the fungal mat, but it is quite difficult in case of the bacterial culture. After producing the microbial enzyme, the purification step is very important, and in addition, it is very complex also, because when we are using these enzymes, microbial enzymes, they should be in pure form at very low concentration, should have the high rate of conversion of substrate into product. So that's why the purification may play an important role in microbial enzyme production. So the purification, in main steps involved in purification are the preparation of the concentrated solution by the vacuum evaporation at low temperature or by ultrafiltration. They'll be concentrating the solution of the enzyme by vacuum evaporation or at low temperature, because enzymes are temperature sensitive, or by ultrafiltration. Then another method is clarification of the concentrated enzyme by a polishing filtration to remove other micro. So clarification will remove all the other microorganism. The another step is addition of the preservatives or the stabilizers, for example, calcium salts and sodium chloride, sugar, alcohols, sodium benzoate will be added in order to preserve the or to maintain the stability of these enzymes. Then another one is precipitation of enzymes with acetone, alcohols, or organic salts like ammonium sulfate or sodium sulfate will be done to increase the precipitation of the enzyme. Drying the precipitate by free drying, vacuum drying, or the spray drying will be done to concentrate the pure enzyme. And later it will be packaged for the commercial supply. So like this, the purified enzyme concentrate will be increased in the microbial enzyme production for employing in the various processes. So these microbial enzymes should be having high rate of conversion of substrate into product at very low concentration. At the same time, they should be very pure. So in this session we learned about what are microbial enzymes and how the microbial enzyme production takes place. That involves isolation of microorganisms, strain development, and inoculum preparation. Further, how we formulate the media for the production of the microbial enzyme, and we concentrated on the different methods of microbial enzyme production by surface fermentation, submerged fermentation, and then continuous culture. And at the end we concentrated on the purification of the enzyme, which is very important step in producing the pure form of the microbial enzyme. I hope you understood this session. Thank you.